Good evening, everyone. This is Dr. Abhijeet, and today I will be discussing the nucleic acids and the nucleotides biochemistry part 2. So, the Oracle has initiated a 90 days foundational live class for university exams, and I will be revising the part 2 today in a very quick, crisp and concise manner. Right, I will be revising it in the form of Q&A, the question and answer format. Right, I will be revising the important topics in the form of question and answer, where I will be drawing the diagrams for you. Right, apart from that, the ones who are appearing for the prof exam, obviously it is for them only, but the ones who are going to give the neet pg upcoming neet pg exam, they can also benefit from this particular lecture. Right, now, okay, I will be discussing today's part in like three pattern of questions. The one, it is the long essay, which I have discussed previously also. The short essays, which consist of 8 and 5 marks, basically. So my main focus will be on the DNA replication today. I will be discussing the steps, the enzymes, the semi-conservative nature, everything. Apart from that, I will be like focusing on the short questions also, the 3 and 2 marks question, right? So that is the point of discussion for today. Apart from that, you already know about the Oracle app. We have the recorded classes, guys. Once you like miss out the live sessions, you can always go on for the recorded version that is available on the app. The live classes we are conducting daily between 6 to 10 p.m. Okay, even the practical sessions, guys, are being recorded. No app will not provide you with the recorded practical sessions. This app is containing all the practicals, whether it is anatomy, physio, biochem, everything is being recorded, right? So, yes. Apart from that, we have the express exam module, guys, where no 1 month before you cannot go on for the textbook to read anything. So everything is covered just under 20 hours, right? So all the important topics are covered in a very crisp, concise, and conceptual manner in the express exam module series, where you can just go on revising like a month before the exam. And the most important part is the hard copy book set, which is available at your doorstep right now, right? It is curated by the expert team, and everything is in the form of Q&A, where they covered the last previous 10-year questions from various university exams. All the important topics are covered in the form of like points. We have riddles also, puzzles also, questions also, and yeah, the notes, the colored notes. So we have the recorded classes, we have the live classes, we have the express exam module where everything is covered in the 20 hours, right? We have the doubt clearing session where you can book, no, you can contact the management team and you can book for your particular doubts if you have, right? So one-to-one doubt clearing session is also available. Apart from that, we have the hard copies book set where you can just go on for the, yeah, the notes, which will be delivered to your home, right? Apart from that, we have the question bank, which is curated by the expert team. It consists of basic questions to the advanced level. Basically, we are covering the entire topic, okay, in the form of these basic questions to advanced question. All the previous NEET PG questions, INI CET questions, everything is covered over here, right? So, yes, and even the clinical vignettes nowadays, the examiner is more focusing on the clinical aspects. So basically, the clinical vignettes are also covered over here, right? So we have the practical sessions which are being recorded, and this is the basic subscription plan, guys. You can go on for it. We have the foundational plan, advanced learning plan, complete learning plan, and the ultimate MBBS plan. And you can always use my code Dr. Abhijeet 20 for additional discounts. So before coming on to the first question, let us try to discuss the very important topic for today, that is the DNA replication, guys. I will be discussing the entire DNA replication in okay. So I will be discussing it in the form of a 10 marks question today, right? Where I will be covering all the essential points that are no required to write on this replication. Now, what is meant by the DNA replication, guys? So answer. This is a very, very important question, guys. It has been asked in various university exams, in the prof exams, multiple times, and it has never been asked less than 10 marks or 8 marks question. So please try to focus this. I mean, it is very, very important. So first, you should know what is the definition of the replication. Definition. Now, The definition says that it is the process of formation. It is the process of formation of two daughter DNA which are each identical to the parent DNA, which are each identical to the parent DNA. Now, what does this mean? Now, let us suppose that this is a cell. Okay, this is a cell. Okay, or I can simply say a parent cell. Now, this parent cell, it is containing two DNAs. I mean, one DNA, it's a double stranded DNA, right? Now, what will happen if the mitosis will take place? There will be two daughter cells which will be formed. There will be two daughter cells which will be formed. Daughter cells which will be formed. Now, what will happen? One of the strand from the parent cell will go to one daughter cell, and one of the strand of the DNA of the parent cell will go to the another daughter cell. So it is the process of formation of two daughter DNAs which are identical to the parent DNA because they are containing the same DNA, right? So what is the concept behind this? What is the concept behind this? We get a replica. We get a replica of parent DNA. We get a replica of the parent DNA in the daughter cell. In the daughter cell, we get a replica of the parent DNA in the daughter cell. Okay, that is the basic concept behind the replication. We already know about the central dogma of the molecular biology. Okay, if I just want to tell you something extra about this, you already know the central dogma of molecular biology, or I will say the modern central dogma of molecular biology, which states that from DNA, what is made? RNA. And from RNA, what is formed? Proteins. From DNA, DNA is made. That is why it is called as the modern central dogma. And that is something called as replication. Replication. From a cell, basically the DNAs are copied, right? From DNA to RNA, and especially the mRNA, it is called as transcription. And from mRNA to protein, it is called as translation. So our point of discussion is from DNA, we are making DNA only. We are making DNA only from the parent. Basically, the daughter cells are containing the DNA only, right? So that is the concept. Now, what is the importance, guys, over here? Remember, it helps in. Now, you can very well see if parent DNA is going to the daughter cell. So what it is passing, guys? It is passing the genetic information. So remember, guys, it helps in passing the genetic information. It helps in passing the genetic information to the progeny. It helps in passing the genetic information to the progeny. Okay. Basically, the base sequences. Now, if it is a DNA, you already know it has base sequences, right? So another definition in the book, it is also given that it is basically the process by which the copying of the base sequences, AGTC, base sequences present in the parent strand to the daughter strand, right? So that is again replication, right? That is again replication. Now, what are the silent features of this DNA replication? Let us discuss the silent features of DNA replication. Now, remember, guys, the DNA replication, very, very important. It takes place in the S phase of cell cycle. Very, very important. Even it has been asked as a 1 mark MCQ also. It takes place in the S phase of cell cycle. Right? You already know, if we just draw the cell cycle, guys, we have G1 phase, we have S phase, G2 phase, and we have mitosis. Mitosis. So DNA replication basically takes place in the synthesis phase, right? S phase of the cell cycle. Apart from that, guys, remember that both the strands of DNA, act as a template. Both the strands of the DNA act as a template. Let me draw for you guys. Now let us suppose that this is the DNA, guys. Right? This is the DNA. Right? One direction is from 3' to 5', and other direction is from, yes, 3' to 5', like this. Right? So both the strands, guys, will act as a template, right? For both the strands, new DNA can occur. Okay. So like that you see in the parent cell, we have two strands. So one strand is copied to one daughter cell, one strand goes to one daughter cell. And in both the daughter cells, we have one strand, and that one strand that is coming from the parent cell, they all are templates. From this, we can synthesize more also, right? So yes, so this is basically acting as a template strand. This is basically acting as a template. Okay, this is also template. This is also template. And from this, we can make a new strand of the DNA, which is highlighted in the red color. So this red color is nothing but, remember, this is the new strand which is formed, or new strand basically in the daughter DNA. Daughter DNA, right? Daughter DNA, right? Now, the third one, remember, half of the parent strand is conserved in the daughter, okay, in the future. Now this is something called as the semi-conservative nature. Semi-conservative nature of DNA replication. Semi-conservative nature of the DNA replication, and this is basically explained by or proved by the experiments that you have already done. It is proved by the experiments by Messelson and Stahl, right? These experiments we have already done in +2, right? Messelson and Stahl, right? So yes, so that is you already know. If I just wanted to draw this, guys, you can just see if this is the parent cell and we have the two daughter cells, right? What is happening? The parent cell is having the double-stranded DNA. One strand will copy to one and one strand will copy to one. And obviously, the new strand will be synthesized accordingly. So both of them acting as template also. And remember that only one strand is copied to one cell. So it is semi. Semi means half. Only one strand is copying to one daughter cell. So it is conserving only half of the strand. So it is semi-conservative in nature. It is semi-conservative in nature. Okay. Now, guys, again, it is following the base pairing rule. So the question also says that the semi-conservative replication, so that I have already explained right now, this is the semi-conservative nature. You also have to know that they all follow the base pairing rule. The base pairing rule. Now, base pairing rule, you already know, adenine binds with the thymine with 2 double bonds and guanine with cytosine with three double bonds, right? So that is the base pairing rule. That is the base pairing rule, right? Even guys, one more thing that you have to remember that, yes, the new strand is synthesized in 5' to 3' direction. New strand is synthesized in 5' to 3' direction. For example, if this is the parent DNA, I'm just taking the DNA out of the cell, and if this is the parent DNA running from 3' to 5' and 5' to 3', like this, they are like parallel, right? So obviously, yes, if one strand is 5' to 3', and half which will be copied over here, it will be 3' to 5'. So the new strand which will be synthesized, guys, always remember it is always in 5' to 3' direction, right? Always in 5' to 3' direction, always in 5' to 3' direction, right? So the new strand which is synthesized, it is always in 5' to 3' direction, right? That is one of the, yeah. Apart from that, you can also remember, guys, that the DNA replication is bidirectional. It is bidirectional. Now, guys, you already know, if we just take a strand over here, that is running from 5' to 3', okay? So obviously, one strand will be synthesizing the DNA in 5' to 3' direction. And we also have another strand simultaneously. Right? It will synthesized in 5' to 3' direction. So basically, the DNA replication is taking bidirectional. So this is yes, bidirectional. Both are in opposite direction. Yeah, bidirectional, right? Bidirectional, right? Now, apart from that, one more thing that you can remember that yes, the DNA replication is semi-discontinuous in nature. Semi-discontinuous in nature. Now, guys, you already know we have the two DNA strands that is in the form of a replication fork, 5' to 3', and this 3' to 5', right? One strand which is synthesized, it is synthesized continuously from 5' to 3'. Okay, this is synthesized continuously. Okay, and this is called as the leading strand. Leading strand. The other one, it is synthesized in the form of these Okazaki fragments, which I will be telling you. Okay, and that is again from 5' to 3', right? So it is synthesized discontinuously. It is not synthesized in a very single direction in a very linear fashion. It is synthesized in like pieces, right? It is synthesized in fragments. It is synthesized discontinuously. Okay, that is the lagging strand. Okay, and that is the semi-discontinuous nature. Semi-discontinuous nature. Okay, it is the semi-discontinuous nature, right? So that is you have to remember all the 7 important like silent features of the DNA replication. The silent features of the DNA replication, right? We have discussed the concept, the definition, right? We have discussed the silent features. All the 7 are important one. Now coming on to the question also says the steps of the DNA replication. Okay, the process, the steps, and the enzymes. Right, I'll be discussing the steps and the enzymes in the same, right? So just put the heading as the steps of DNA replication. Steps of DNA replication. Steps of the DNA replication. Now, over here, we have like 5 steps, guys. Just try to mention all of them. Then we'll discuss it one by one. The first one, it is the identification of origin of replication. Identification of the origin of replication, that is called as ORI. I'll be discussing each step in detail, no need to worry. Then we have unwinding of DNA. unwinding of DNA. Then we have formation of replication fork. Formation of replication fork. Then we have DNA synthesis. DNA synthesis, that is the leading strand and the lagging strand. Lagging strand. Then we have the termination part. So these are the 5 steps under which we are going to cover the entire DNA replication. Now let us discuss it one by one. Now, the very first one that we are going to discuss, it is the identification of origin of replication, that is ORI. Identification of origin of replication. Now, what happens? let us just suppose that we have a bacterial DNA. Okay, we have a bacterial DNA, and bacterial DNA are double standard circular, right? So we have bacterial DNA, and we are taking it as double standard, and this is circular, right? Now, in this DNA, guys, we have a region over here. We have a region over here, and this region, it is called as AT rich region. This is called as the AT rich region. Now, when I say AT rich region, I'm telling adenine and thymine are most common in this region. That is called as the AT rich region. A basically stands for adenine, and T basically stands for thymine, right? Now, guys, over here, we have something called as the origin of replication. Okay, we have origin of replication. Now, this is basically what does this mean? It is the fixed point of DNA where replication begins. It is the fixed point of DNA where replication begins. Right? Now, now there is a protein, guys, that is called as the ORIBP. Now, it is called as the origin of replication, right? ORI binding protein. It is called as ORI binding protein. Now, what will happen? This protein will go and bind to this ORI. Okay, this protein will go and bind to this ORI. Now, what will happen? Now, after this ORI binds to this after this ORI BP, that is the origin of replication binding protein, binds to the ORI. Okay, let me again draw it, guys. Okay. Now, what will happen over here? We have this, yes, the origin of replication. And to this, the green color one, that is the binding protein, comes and bind. Okay, what will happen? The AT rich region, the red color region, will start unwinding. It will start unwinding. Okay, the AT rich region unwinds. Okay. Now, this region is very no susceptible for denaturation also. It is susceptible for denaturation also. denaturation also, because it is, yeah, less temperature. Guys, why AT rich region? Why not GC region? See, guys, it is very easier to break a bond which is containing only two double bonds. A and T are bounded together by two double bonds. G and C are bounded together by three double bonds. It is very, very difficult to break a three double bond rather than breaking a two double bond. So the AT rich region will unwind only when the origin ORI binding protein will come and bind to ORI. Then it will give the signal and they will unwind with each other. Okay. They will unwind with each other. Now over here comes the rule of one important, okay, protein over here. And remember, guys, the protein over here that is very, very, very important. It is called as the single stranded binary or binding proteins. Single stranded binding proteins. Very, very, very important. Now, what is the function of these proteins? These two strands, which are no apart from each other, can also come together to each other. Okay, they can also come together to each other. Okay, again. So basically, what does these proteins do? They prevent the re-annealing of the separated DNA. They prevent the re-annealing of the separated DNA. Re-annealing का मतलब है, it means that it will not allow the separated DNA to come closer to each other, right? So again, only then will no the replication will take place, right? Now, in humans, in books, guys, what you will read, in humans, basically, we give a very special name to these proteins. Okay, in humans, basically, these are given as special names, and this is also called as replication protein A. It is also called as replication protein A. It is also called as replication protein A. So that is the identification of the origin of replication. Okay. Now, again, two things that you have to take away from this slide is key in eukaryotes beta. We have prokaryotes and we have eukaryotes. Now, what is the difference, guys? In both the cases, in the replication, origin of replication, only single origin of replication is there in prokaryotes. Okay. Whereas in eukaryotes, we have multiple origin of replications. We have multiple origin of replication. This is the very first difference between the replication of prokaryotes and eukaryotes, right? So we have a single origin of replication in prokaryotes and we have multiple origin of replication in prokaryotes, right? So that is the first step. That is the identification of the origin of replication, identification of the origin of the replication. So we have done the very first step, identification of the origin of replication. The second one, it is the unwinding of the DNA, unwinding of the DNA. Let us just try to write this. I hope Dharanika, you have done this. Just give me a thumbs up in the chat box if it is done, Dharanika. If you have drawn this in the paper, also you have to draw in the same way. Okay, very good, beta. Very good. I hope you are understanding, Dharnika. Now, coming on to the second one, that is the unwinding of DNA. Very good, Peter. The second one, that is the unwinding of DNA. Now, unwinding of DNA. Now, they have already started unwinding. Unwinding means they are started getting separated. Now, further unwinding of DNA is done by, okay, in the prokaryotes, basically by an enzyme, very, very important. Again, I'm taking an enzyme over here that is called as helicase. Helicase. It is ATP dependent enzyme. ATP dependent enzyme. So remember that this helicase in prokaryotes will help in further unwinding of the DNA. But remember in case of eukaryotes, we have MCM. MCM. Now this MCM stands for mini chromosome maintenance complex. Maintenance complex. Mini chromosome maintenance complex, right? So it is like different in both the cases. Unwinding will take place. Unwinding was initiated earlier. Now unwinding will take a greater speed and it will further unwind rapidly. And in prokaryotes, it is the helicase. In eukaryotes, it is the mini chromosome maintenance complex enzyme, right? Coming on to the third step, guys, the formation of the replication fork. The third one, that is the formation of the replication fork. Okay, now what happens? Just try to see over here. Formation of replication fork. Now, what happens? Now, now guys, remember this is the replication fork. Each half, basically what will happen, guys? The replication fork will be formed. Replication fork will be formed, right? So basically what is actually this structure is, the structure it is of a replication bubble, right? Now what will happen? How the replication fork will be formed? Let us just try to see in the DNA synthesis wala part. Okay, the fourth part. Let me just draw for you, then you will be like better understand it. DNA synthesis. DNA synthesis. Now what happens? We have from 5' to 3' direction. Okay. And this is from 3' to 5' direction. Right? So this is the parent DNA. This is the parent DNA. Now what happened? RNA primer. RNA primer. What is this RNA primer? Remember, guys, it is about 10 nucleotides in length, and it is made up of ribonucleotides. It is made up of ribonucleotides. Okay, it is about 10 nucleotides in length, and it is made up of ribonucleotides. It is basically synthesized by an enzyme. Again, the enzyme is very, very important. It is synthesized by an enzyme that is called as primase. It is synthesized by an enzyme that is called as primase, right? It is synthesized by an enzyme that is called as primase. Now, what happens? The new strand will be synthesized continuously, right? The new DNA strand It is synthesized continuously. Okay, it is from 5' to 3'. It is from 5' to 3'. Now, guys, remember that in prokaryotes, okay, one enzyme is this. Remember, in prokaryotes, it is done by DNA polymerase 3. It is done by DNA polymerase 3. It is done by the DNA polymerase 3. So that is the leading strand. Okay, that is the leading strand synthesis. Coming on to the lagging one. So it is the leading strand. It is the leading strand. Coming on to the lagging strand, right? No. We have over here, okay, the leading, we have the lagging strand template. Lagging strand template. Now what happens? Basically, we have multiple 5' to 3'. We have multiple RNA primers over here. Okay, we have RNA primers, right? And over here, basically what will happen, the short DNAs will be synthesized. The short DNAs will be synthesized. Short DNA will be synthesized. And these are nothing. These are called as the Okazaki fragments. These are called as the Okazaki fragments. Okay, with the help of DNA polymerase 3. Right? So these Okazaki fragments basically are absolutely okay. If I just want to differentiate, guys, 1000 to 2000 basically nucleotides, they are in prokaryotes. Okay. And 100 to 250 basically they are in eukaryotes. Okay, 100 to 50 they are in eukaryotes, right? So again, basically what happens, basically the primers, the RNA primers over here, they are further removed. They are further removed. And remember, the gaps are filled. The gaps are filled, okay, by DNA polymerase 1. DNA polymerase 1, right? So yes, the gaps are filled by the DNA polymerase 1. Basically, we have the ligase enzyme, which helps in sealing of the nicks, right? It acts like a molecular fevicol, right? It helps in the connection of these nucleotides, the sealing of the nicks, right? So ligase also basically, the DNA ligase also requires ATP. The DNA ligase also requires ATP. Okay, so basically the primers which were there in the starting, it is removed. The gaps are filled by DNA polymerase, and further they are stick to each other with the help of enzyme DNA ligase. Further they are connected together with the help of DNA ligase. Coming on to the last one, the last step, that is the termination. That is the termination. Okay. Now in prokaryotic DNA, guys, you just have to mention over here in prokaryotic DNA, basically there is a site called TER-A, right? TER site, basically it is a conserved sequence. Okay. And yes, it is a conserved sequence, and it is basically the the termination part, okay, the termination utilization sequence. And over here, yeah, we have a site, and over till that part, if it is reaches, it is basically not terminated, right? So it is a conserved sequence, okay. And yes, the unwinding basically stops at this particular sequence. So that is regarding the steps of the DNA replication, which we have discussed completely in detail with the help of all the enzymes also. So we have discussed DNA polymerase, concept of semi-conservative, all the key enzymes, polymerase, helicase, single standard binary proteins, ligase, right? Even we have discussed all the 5 steps, the significance, why for the progeny it is needed, okay, the process, how does it take place, the leading strand, the lagging strand formation, everything is being discussed. I hope you understood this, right? Dharnika, I hope it is clear. Right. So we have discussed all this. Yeah. Now, let us just try to discuss the very first question for today, because we have discussed it in the last class also. So I'll be just going in, yeah, very good, Victor. In quick manner, the other questions, because we have already discussed this in the last class also. Now the question says, describe the biological importance of nucleotides beyond role in nucleic acids. So functions in energy, ATP, cell signaling, enzyme cofactors, and metabolism. Now, guys, you already know the nucleotides are the building blocks of nucleic acid. We have already discussed this in the ATP, guys. ATP is nothing but adenosine triphosphate. Adenosine triphosphate. It is the energy currency of the cell. You already know it is required for muscle contraction, transmission of nerve impulses, and transport of nutrients across membrane. Right? So ATP is required for ligase type of enzymatic reaction. Right? Okay, it is also required for enzymatic reactions, right? For example, ATP, it is converted into adenosine diphosphate, and a phosphate molecule is released, basically the pyruvate carboxylase enzyme. Okay, so basically it is helping in the conversion of pyruvate, okay, to acetyl coenzyme A. Okay, so all the further reactions. It is also no further hydrolyzed to AMP. Obviously, AMP stands for adenosine monophosphate. Monophosphate. Okay, and obviously the two inorganic phosphates will be released, right? So yes, ATP is also involved in phosphate transfer reactions, that is the glucokinase reaction. Now guys, you can see we have three phosphate, mono, di, tri. For example, if glucose, okay, has to convert it into glucose 6 phosphate. Now which enzyme we are using over here? We are using hexokinase. Hexokinase. Now you already know over here the ATP will be used and it will be converting into ADP. Triphosphate will convert into diphosphate. One phosphate will go and attach to glucose. Which position? 6th position. Okay. So it is glucose 6 phosphate. At the 6th carbon atom, the phosphate is added, and this phosphate is coming from the ATP. ATP is broken down to ADP. The phosphate which is released, it will come and attach over here. Right? All, or there are many reactions, guys, that I will be discussing in future. It is involved in pyrophosphate transfer reactions. It is involved in adenosine transfer reactions. Okay, it is also act as a secondary messenger. Okay, whenever I will be discussing the hormonal pathway also, it also acts as a secondary messenger also. Okay, now for it also like have coenzyme roles, right? It has adenosine nucleotides, NADP, flavin mononucleotide. Okay, so that is flavin adenine dinucleotide. Okay, even in the phosphoadenosine phosphosulfate or active sulfate reactions. Okay, apart from that, it is also no acting GTP, guanosine triphosphate. Guanosine triphosphate, it is required for the provision of energy during protein synthesis. Okay, cyclic GMP, that is the guanosine monophosphate, it is a secondary messenger. Okay, formed from GTP only. Obviously, GTP triphosphate will be broken down into di, then it will be converted into monophosphate. Okay, by the help of enzymes guanylate cyclase. Okay, we also have cytidine nucleotides, which is required for the synthesis of lecithin. We have already discussed the phospholipids, right? The phospholipids over here. Even they are also required for the synthesis of cephalin, right? Phospholipids are the major components of the cell membrane, the L by S ratio that we have discussed. It should be more than 2, which indicates the fetal lung maturity, right? Apart from that, it also helps in the like the uridine nucleotides. Basically, it's a carrier of glucose in the synthesis of glycogen. Obviously, glycogen, it is the storage form of glucose in humans, right? UDP, glucuronic acid, it is basically required for detoxification of bilirubin also, right? So that was regarding the first question. We have already like covered it in the last session also. So it's again repeated, right? We have discussed the DNA part also. Now the messenger RNA also, guys, we have already discussed in detail in the last session also. But over here, you just have to remember that the mRNA is synthesized from the template strand of DNA. Now you understand the DNA has two strands and both the strands are acting as template. And from one template, basically the mRNA is synthesized, and that is something called as the transcription, right? So the genetic message which is there in the DNA, it is transcribed to mRNA, right? So yes, also remember that the mRNA acts as a template for protein synthesis also, for DNA to mRNA and from mRNA to proteins, right? So you already know we have 5' end and the 3' end, right? 5' end has start with 7-methyl GTP, and 3' end it is having a polyadenylate tail of 22 2 to 250 AMP residues, right? What is the function? It helps in protein synthesis. Okay, it comprises of codon, the three bases triplets, right? The A, G, C, and U, because it is RNA, so uracil will be there. There are like 64 possible codons. 61 are for coding amino acids. 3 codons are referred as nonsense codons, right? So that is something you have to remember. The nonsense codons are UAA, UGA, and UAG. Now, hydrogen bonding, guys, this question we have already discussed in detail in the last session also. You already know the adenine binds with the thymine and guanine binds with the cytosine with the 3 hydrogen bonds, and adenine binds with 2 hydrogen bonds. Obviously, they are providing a structural stability to the structure, right? Central dogma we have already discussed, guys, from DNA. What is made DNA? That is called as replication. From DNA, what is made mRNA? That is called as transcription. And from mRNA, what is made? Proteins. That is called as translation. That is called as translation, right? So you have to describe transcription and translation and replication in detail. rRNA we have just discussed. Yeah, the rRNA we have already discussed in the last class also, right? Now, this rRNA basically helps in the protein synthesis, right? Protein synthesis. Now, remember, guys, they are the structural components of ribosomes, right? So they are made up of two subunits. They are made up of two subunits, large and small. Okay. So remember in eukaryotes, basically the smaller subunit, it is made up of 18S. The large, it is made up of 28S, right? Prokaryotes, it is the small one, it is made up of 16S. The large basically consists of 23S or 5S, right? So apart from that, guys, they also have catalytic role. Okay. So the ribosome basically it acts as a ribozyme also. It acts as a ribozyme also. Okay. The RNA molecule with the enzymatic activity. No, even the peptidyl transferase center, guys, you already know of the ribosome. It is responsible for catalyzing one reaction or for the formation of the peptide bonds between amino acid. It is mainly done by this rRNA, right? It is done by the rRNA, right? So this is all which you can mention. And that was about today's session. Thank you for your patience and listening me out. Thank you.